Question number 12 show us two types of the gene in the tomato. So as we can see here, first one is the shape, yeah, the shape of the tomato. So if it's, let's say it is a round, it is dominant. Round would be dominant, so it's a big R. Okay, then for the oval, oval would be the recessive, small r. Well, another gene would be the fruit that is smooth or rough, yeah. So if let's say it is smooth, then it will means that it will have dominant, big S, and then rough would be recessive, small s. Okay, so now you can see that the test cross is done and they give us the number of the offspring. Okay, so the number 130, 1270, and then 1310 and 110. Okay, so when you look at the number of the offspring, so first thing first is we list them down first. Yeah, we will list them down. So there are total 130 round smooth. Then we have 1270 of the oval smooth. And then 1310 of the round rough. And lastly, 110 of the oval rough. Okay, so once you have the phenotype of all the offspring, okay, normally I would also write down the genotype. Yeah, I will write down the genotype, the possible genotype as well. So for the round smooth, you can see that it will be basically 2 dominant. Okay, that means that at least one R gene that's dominant, at least one S gene that's dominant, then only then it will be round smooth. Okay, so that is that when the both RS gene are both dominant. Okay, then next would be the oval smooth. Oval smooth, it will be small r. Okay, and then smooth would be at least one big S. Then next would be round rough, yeah, round rough. Round rough would be one big R at least, and then rough would be small s, small s. All right, and then lastly is homozygous recessive, where small r, small r, small s, small s. Right, so this would be some key that allow us to predict, yeah, allow us to predict the genotype of the parent actually. Yep. So done with this, then next is of course we have to find the ratio based on the number of the offspring. Yeah, based on the ratio. Okay. So to get the ratio, we will divide by the smallest number here, which is 110. So we will divide everything with 110. Okay. So let me divide. Okay, so let me get the calculator. So 130, if let's say you divide by 110, okay, so it will be around 1 point something, right? So yeah, the ratio will put it as 1 first. Okay, so 1270 divide by 110. Yep, same thing again. So it's around 12 point something. So the ratio is around 12. Same thing goes to round rough. 1310 divided by 110. So again, it's 12 point something. All right, then lastly is 110, then you get 1. So from the ratio, you can see that it's 12 to 12 to 1 to 1. Okay, so when you see there's a big difference in the ratio, then again, we can suspect that this might be the link gene. Yeah, this might be the link gene. Okay, and given that the ratio is so huge in the difference, right? So the 12 to 1, yeah, that's a very big difference in the ratio. Yeah, so most likely this is actually the link gene. Okay, so that's why for these questions, we can say that this is actually deviation. Yeah, this question should be deviation, and it's related to the link gene, right? Related to the link gene. And how to solve the questions that is related to link gene, we will always use the type 2. Yeah, the type 2. Type 2 method is still the same. Yeah, that means you still find the first step, still find the genotype of the parent. Then after that, second step, find all the possible gamete, right? All the possible gamete of the parent can produce. Okay, and then third step, there's one more step for this type 2 method, yeah, is that you have to find which one is parental, which one is the recombinant. Okay, then lastly, only yes, the type 4, the step 4 would be finding the phenotypic ratio or the genotypic ratio. Okay, so that is basically the type 2 method. Okay, so done with this already, then next we will do the same thing again. First step, find the genotype of the parent. Yeah, find the genotype of the parent. Okay, so based on here, it's a test cross. A test cross carried out on a round, smooth plant. Right? Round, smooth plant. Yeah. Okay, so that means that this parent, it must be at least one big R, at least one big S, because it's round, smooth. Then it is crossing with test cross, right? So test cross will mean that yes, you have to cross it with the homozygous recessive individual. Okay, so based on this, yeah, based on this, yep, that would be the parental genotype. Okay, the parental genotype. Okay, but how are we going to predict this one? Yeah, how we are going to predict this one? Okay, so based on the ratio here, yeah, based on the ratio here, first thing first, yeah, if let's say it's a linked gene, right? So we all know that there are different ways to write that. Okay, so if let's say it's a heterozygous, yeah, then basically there are two ways to write that. So it's R big S slash, okay, then the R or S, okay, or it can be big R small s, then small r big S. Okay, so you might wonder why I directly write that this is a heterozygous. Okay, so the reason is because when we do the test cross, right? Okay, when we do the test cross and you find out that there are, yeah, they are the individual, the phenotype, okay, or genotype, yeah, that is homozygous recessive. That is homozygous recessive, yeah. Whenever you doing the test cross, and then in the offspring, that is the offspring that with the genotype that is homozygous recessive, then normally the parent, another parent that's being tested must be heterozygous. Because if without the heterozygous, it's almost impossible for us to get this, yeah, in order to get this homozygous recessive individual here. Okay, so that's why, yeah, whenever you see that is whenever there is a test cross and then this is being produced, this type of genotype is being produced, then normally it would be the heterozygous for both. Okay, so that's why, yep, we can predict that. Okay, so second, this heterozygous genotype, it should be like this or the second one. Yeah, we are not sure which one, right? Okay, so here's another hint that can help you to predict. Okay, so over here you can see that, yeah, for those homozygous dominant one, for those both dominant trait, for those that's both dominant trait, if let's say the number is smaller, okay, compared to those individual, those offspring that have only one dominant trait. You can see here, right? This one only big S. This one only one big R. Okay, so that means that only one trait is dominant. Only one trait is dominant. Okay, so if let's say the offspring with only one trait is dominant is having larger ratio, then that will mean that the genotype of the parent, yeah, the genotype of the parent will be the second one. If let's say the the offspring, yeah, if let's say the offspring with both the R and S also dominant trait is having higher ratio, then this will be the parent the parent genotype. Okay, so it is either the one with the both dominant trait and both recessive trait is having higher ratio, then we will choose this one. But if let's say it is the offspring with only one trait, one dominant trait having higher ratio, then we will choose the second one. Okay, so done with this. Then next, we will cross with the homozygous recessive individual because it's a test cross. So this one, I think is very simple to write that. Okay, so now, yep, we assume that this is a link gene. Okay, so that's why we have to use the second method. Okay, so in the second method, now we have the parent genotype already. Next is we can actually figure out what are the possible gamete. Okay, what are the possible gamete here? Okay, so I think I have taught you all before, right? For the link gene, how we handle this is basically we'll draw two lines. Okay, so to indicate, okay, for these two this R gene and the recessive S gene, they will be on the same chromosome, and then this will be the on the second one. Okay, so the parental would be the big R, small s, and the small r, big S. This would be the two parental gamete. So we'll write that down. Okay, so this would be the parental. This would be the parental. Then crossing over will occur. So big R, big S, and then crossing over will happen. So it's a small r, small s. Okay, so these two would be the recombinant. Well, for this individual, it is going to produce only one type of the gamete. Yeah, this parent, yeah, only produce one type of gamete only. So which is small r, small s. Okay, so done with this, then next we will do the cross. Okay, so done with the cross. Now we know that these two are parental, these two are recombinant. Okay, so based on the genotype, try to link it to the phenotype. Yeah, try to link it to the phenotype. Okay, so this one contain one big R, so that means it must be round. Okay, and then two small s. So two small s, it must be rough. Yeah, so round and rough. Yeah, round and rough is here. You can see this round and rough. Yeah, okay, so round and rough, the number is, yep, 1310. Yeah, 1310. So it's 1310. Then next, the second one with two small r and one big S. So two small r would be the uh over. Yeah, the over. Yeah, so one big S would be smooth. So this one would be this one, over smooth. Yeah, it will be the over smooth. Okay, so total number is 1270. So 1270. Okay, then next, one big R, one big S. Yeah, one big R, one big S. It must be round smooth. Round smooth is here. Okay, so 130. That's the total number. And then lastly, over rough would be the number 110. Okay, so once we have the number, the next step is, if you want, you can try to calculate the recombinant frequency to further confirm that this is a link gene. Yeah, to further confirm that this is a link gene. Okay, so I will use a blue color to show that. Okay, but in fact, this one you don't have to show in your working. Yeah, so it's just for you to check whether, yep, this is actually a recombinant frequency. So recombinant frequency equals to the two recombinant type, which is 130 plus 110. Okay, so we divide by the total number of all. Yeah, total number of all. So total number of all are 1310 plus 1270 plus 130 plus 110. So total up, it will be 2820. So 2820, we times 100%. Okay, so what we get is around 130 plus 110 divided by 2820. So we get 8.5%. Okay, so it is 8.5%. It is lesser than 50%. Then that means that, yeah, we now can confirm that the R gene and S gene, they are the link gene. So if they are the link gene, when we write the genotype for this parent, because the question is asking the genotype of the round smooth parent here. The test class is carried out the round smooth parent, right? So yeah, what is the genotype of this round smooth parent? So round smooth parent. Okay, so we'll write that down. So genotype of the round smooth parent. Okay, so it will be equals to this one. This should be the genotype. So it will be the big R small s slash small r big S. Okay, so with that, yes, that's all. So we will write like this because in the link gene, right, in the link gene inheritance, right, it is more specific to write like this. Okay, since they are asking for genotype, so yeah, better we give it that is the genotype that's specific. Okay, so yeah, that is all for this question number 12.